Welcome to the debate. When we think of writing, we picture, ink on a page or maybe fingers on a keyboard. But imagine a machine that writes entirely new books, books that actually leap off the page, replicate themselves, and, well, walk around the room.
Right. Which sounds like science fiction.
Exactly. But on August 6th, 2026, we stopped having to imagine it.
Yeah. Because the language this machine learned wasn't English or Mandarin. It was the foundational code of life itself, DNA.
Spot on. Researchers from Stanford University and the Arc Institute published this landmark study in the journal Science. They detailed how an artificial intelligence model named Evo2 designed 16 fully functional, never-before-seen viral genomes. And it did this completely from scratch.
Which is just wild to think about.
It really is. So today, we are examining the core tension of this breakthrough. Does the open source release of generative AI models capable of writing functional viruses, represent a necessary leap forward in solving the global antibiotic resistance crisis? Or, on the flip side, does it constitute an unacceptable dual-use biosecurity risk?
A massive risk, yes.
Well, I argue that open sourcing this model is a vital, manageable step for medical innovation. It gives humanity a crucial, necessary defensive advantage against superbugs.
And I argue that distributing a generalizable AI capable of generating novel viral genomes completely outpaces our current regulatory frameworks. It presents an unprecedented, severe dual-use biosecurity threat, one that we are fundamentally unprepared for.
Look, if you're listening to this, you might be thinking, why on earth would anyone want to generate novel viruses?
It sounds like a supervillain plot.
Exactly. It sounds insane. But you have to look at the massive crisis we are losing right now. According to the World Health Organization, we are literally running out of effective antibiotics. Bacteria are evolving rapidly. They're surviving drugs that have worked for decades.
Right. I mean, the traditional medical arsenal is definitely failing. Pathogens like MRSA and drug-resistant tuberculosis are killing millions. It is a terrifying reality.
Right. And the best alternative we have to antibiotics is phage therapy. We use bacteriophages, which look almost like these microscopic lunar landers.
They do. They look totally alien.
They really do. And they are specialized viruses that only infect and kill specific bacteria. But the problem is that historically, finding or engineering the right phage for a specific resistant bacteria has been painstakingly slow.
Yeah, it's artisanal work.
It is. A patient is dying of a superbug, and scientists are spending months hunting through soil samples or manually tweaking genomes trying to find a phage that works.
It's a needle in a haystack problem.
A global haystack. But what Brian Hie, Samuel King, and their team at Stanford did was use Evo2 to fundamentally alter that timeline. They started with a highly studied bacteriophage called Phi X-174.
Which has a tiny genome, right?
Super tiny, just 5,386 nucleotides, 11 genes. Evo2 generated about 700,000 potential variants of this virus. But here is the critical part. The AI didn't just copy and paste existing genetic sequences.
No, it hallucinated new ones.
Well, the 16 viable viruses that resulted contained between 67 and 392 entirely new mutations. Mutations not found anywhere in nature.
And they actually functioned.
They functioned perfectly. By successfully automating the design of these novel genomes, this technology offers a scalable way to generate diverse cocktails of phages.
I hear you, but
Wait, just think about the impact. If a superbug mutates to resist one phage, we can instantly generate 10 more that it has no defense against. We finally have a viable pathway to defeat multi-drug-resistant pathogens. This is a profound medical breakthrough that we desperately need.
okay, but the reason it can build those phage cocktails so fast is exactly what terrifies me. It's not just copying known cures. It has learned the underlying grammar of life itself. And, grammar can be used to write anything, including a weapon.
Ah, so you're talking about the dual-use risk?
Yes. Dual-use research of concern. Look, from a computational biology standpoint, Evo2 is a marvel. I'll give you that. But we have to look at how this model actually operates. It functions very much like a text-based large language model.
Like ChatGPT.
Exactly. But instead of predicting the next word in a sentence, it predicts the next nitrogenous base, the A, C, T, or G in a DNA sequence. Just as ChatGPT knows that the word peanut is usually followed by butter, Evo 2 knows that a specific sequence of A, C, T, G will fold into a protein capable of penetrating a cell wall.
Sure, because it was trained on an immense data set.
Right. It was trained on the genetic sequences of millions of organisms, viruses, bacteria, plants, and humans. It understands how biological components fit together across the entire tree of life. I look at the analysis from Moritz Henke at the Johns Hopkins Center for Health Security.
He's a sharp guy, yeah.
He is. And he points out that if a generalizable genomic model can learn the grammar to write a harmless bacteriophage, that same underlying capability could be manipulated by bad actors to design dangerous, novel human pathogens.
But that's a massive leap.
Is it? The medical promise is real, sure, but distributing the digital blueprints to create living viruses to anyone with an internet connection is a massive vulnerability.
Okay, hold on. We need to separate the digital generation of a sequence from the physical creation of a virus. The physical world acts as a massive friction point.
Not as massive as it used to be.
But it's still there. Look at the empirical reality highlighted by researcher Garcia Ojalvo in response to this exact study. Look at the Stanford experiment's numbers. Out of 700,000 AI proposals, they computationally filtered it down to 285 candidates to actually synthesize chemically in the lab.
And out of those, only 16 actually worked?
Exactly. Only 16 yielded viable viruses that could successfully replicate and cause lysis. The actual bursting and destruction of the bacterial cell. That is an incredibly low success rate for a team of world-class scientists in a state-of-the-art lab.
But they still did it.
Yeah, but giving someone a blueprint for a complex nuclear reactor doesn't give them the enriched uranium or the facility to build it. Aren't the physical requirements of biological synthesis, specialized lab equipment, chemical precursors, the need to insert synthesized DNA into a host cell, aren't those a sufficient safeguard against immediate misuse?
Come on. Relying on the friction of the physical world is a losing bet. Generative AI drastically accelerates the exploration of the genetic space. Synthetic genomics expert Patrick Hie pointed this out perfectly.
What did he say?
Before Evo 2, if a bad actor wanted to engineer a pathogen, they had to understand the incredibly complex interactions of genomics, protein folding, and cellular biology. Now, the AI does the complex reasoning for them. The hardest part of biological engineering, the actual design, is now automated and freely available.
But they still can't just print a virus on their home inkjet printer.
Not an inkjet printer, no. But the democratization of biotechnology is advancing almost as fast as AI. We are seeing the rise of benchtop DNA synthesizers. The cost of genetic synthesis has plummeted.
True, but commercial synthesis providers exist all over the world, and they screen their orders.
Yes, they screen orders for known dangerous sequences, like Ebola or smallpox. But how do they screen for a completely novel AI-generated sequence that has literally never existed in nature?
They screen for homology. They compare it to known pathogen databases.
It completely fails if the AI generates something with 300 novel mutations. A synthesis company's safety algorithm might not flag it at all because it doesn't look like anything in the threat database.
I mean, that's theoretically possible, but
It's highly probable. The physical bottleneck is eroding, and while it might take a rogue lab several tries to get a viable pathogen, the AI has already handed them the 285 most likely candidates out of 700,000. It turns an impossible search into a highly targeted one.
okay, but we are acting as though the researchers just threw all the world's DNA into a blender and posted the results on Reddit.
They kind of did.
Samuel King took highly active, deliberate safety measures. They purposely excluded viruses capable of infecting complex organisms, like humans and animals, from the training data. The architecture was actively constrained to only focus on bacteriophages.
Right, and I question how robust those constraints actually are.
Well, look at what the AI accomplished within those constraints. One of the variants they produced, Evo 536, is fascinating. Human biologists tried to fix this one broken truncated protein in the virus, but they treated it like an isolated puzzle piece, and they failed.
Sure, humans are bad at multidimensional genomic math.
Exactly. But EVO2 looked at the whole puzzle. It realized that if you have a defective stubby protein, you can mutate the surrounding genetic code to physically bend the genome. You alter neighboring proteins to support the broken one.
Like an architectural buttress?
Yes, that is a level of holistic 3D genomic reasoning we just don't have manually. And it applied that brilliance strictly to a bacteria-killing virus.
Okay, you just perfectly articulated why this is so dangerous.
Wait, how?
EVO 536 succeeded where human biologists failed exactly because it understands the holistic context of the genome better than we do. It understands how to compensate for structural deficits by rewriting the surrounding code. Now, apply that capability to a malicious goal.
But it can't be applied to a malicious goal. Human viruses were excluded from the fine-tuning step. The model doesn't have the vocabulary for human pathogens.
That fundamentally misunderstands how foundational models work. The base model, EVO, was trained on a vast array of organisms across the entire tree of life. It learned the general genetic grammar you are praising from that massive dataset.
Wait, walk me through that. If Stanford explicitly stripped out human viruses from the fine-tuning data, how does the model still know how to write them?
Because fine-tuning is just a behavioral guardrail applied at the very end of the training process. Think of it like teaching someone all of human physics, chemistry, and engineering. That's the base model.
Okay.
Then during fine-tuning, you say, only build bicycles. The knowledge of how to build a combustion engine or a missile is still baked into the underlying neural network weights. You've just temporarily instructed it not to use them.
So you're saying the guardrail is fragile?
Extremely fragile. Excluding human viruses from the final fine-tuning step doesn't erase the underlying capability of the base model. If a sophisticated bad actor takes the open-source base model and fine-tunes it themselves on human pathogens, which, by the way, are publicly available in genetic databases, that safeguard is completely bypassed.
I hear you, but that drastically overestimates the autonomous leap from a bacteriophage to a human pathogen. Let's ground this in the biology for a second. Human biology is exponentially more complex than E. coli.
Obviously.
Right. Viruses that target human cells have to navigate incredibly sophisticated immune responses, varied cellular receptors, and dynamic environmental variations. If even Stanford researchers at the cutting edge struggled to achieve a high viability rate with a deeply understood, incredibly simple genome like Phi X-174, the very first genome ever sequenced back in 1977, isn't the fear of generating a complex human pathogen vastly overstating the model's current capabilities?
Today, maybe. But look at the timeline. 2 years ago, AI couldn't design a functionally viable novel protein, let alone a 5,000 nucleotide viral genome.
It is moving fast.
Fast is an understatement. Today, it is writing them from scratch. If we wait for this model to become physically perfect at generating a human pathogen on the first try before we implement strict governance, the game is already over.
So you want to lock it up?
I want oversight. The point that Johns Hopkins Center for Health Security is making isn't that EVO2 is going to cause a pandemic tomorrow. It's that the open-source release of foundational genomic models establishes an incredibly dangerous precedent. We are distributing the foundational grammar of biological design without a functional regulatory net.
Which brings us to the philosophy of open source, and why Hie and King's decision to make EVO2 free to download was deliberate and completely necessary.
Necessary?
Yes, we are in an evolutionary arms race with bacteria. When we use traditional, broad-spectrum antibiotics, we exert selective pressure. The strong survive, multiply, and become superbugs.
Right, the classic evolutionary bottleneck.
Exactly. And even if we use a single, naturally occurring bacteriophage, the bacteria will eventually mutate to evade it. The treatment fails. We desperately need the open, rapid generation of diverse genomic variants to stay ahead of natural evolution.
I agree we need the variants, but
By open sourcing EVO2, thousands of researchers worldwide can start designing bespoke phage cocktails right now. If a superbug emerges in a hospital in Mumbai or a clinic in Buenos Aires, local scientists could theoretically use this open source tool to rapidly generate a tailored viral countermeasure on site.
I understand the instinct to arm the global scientific community. Truly, I do. But open sourcing this technology democratizes both the defense and the risk simultaneously.
You are assuming a symmetrical playing field where the good actors will always outpace the bad actors.
Well, why wouldn't they? There are thousands of legitimate scientists for every rogue actor out there.
Because in biosecurity, defense must be perfect, while the offense only needs to succeed once. Our current regulatory frameworks are entirely stuck in a pre-generative AI era. They are built to monitor the physical transport of known pathogens across borders, not the digital transmission of algorithmic capabilities to generate new ones.
So what's the solution then?
I support the proposal that biological models must undergo rigorous dual-use capability evaluations before public deployment. We need a system where the benefits of genetic genomics can be accessed via secure, cloud-based APIs.
Ah, an API, meaning an application programming interface, essentially a digital checkpoint where researchers can request genetic sequences without actually downloading the whole brain of the AI?
Exactly that. A gated system where the model lives on a secure server. Researchers submit their prompts, the system checks the outputs for dangerous sequences, and then sends the results back. We can monitor what is being generated, rather than letting anyone download the foundational model onto their private offline servers where, frankly, all oversight simply vanishes.
But restricting access to an API governed by a few elite institutions creates a massive bottleneck for global scientific progress. Think about the historical context of the Dirac debate. We've been having this exact conversation for over 2 decades.
And we should keep having it.
Sure. But remember when scientists reconstructed the 1918 Spanish flu virus? There was a massive outcry that publishing the genome would lead to bioterrorism.
Which was a completely valid concern at the time.
But it didn't happen. Instead, sharing that genetic data allowed the global scientific community to better understand viral transcription and prepare for future influenza pandemics. Open science has consistently proven that the collective defense mounted by thousands of legitimate researchers far outweighs the theoretical risks posed by rogue actors. Transparency allows us to build safeguards within the tools themselves.
Okay, but the 1918 flu comparison actually highlights my exact problem.
How so?
When we published the 1918 flu genome, we gave the world the sequence of one specific pathogen. It was a static piece of information. What EVO2 represents is a machine capable of generating infinite novel pathogens. It is the difference between giving someone a bullet and giving them an automated munitions factory.
A munitions factory that currently only makes bullets for bacteria.
For now. And while you champion the collective defense of open science, biological threats don't respect borders or academic intentions. If an AI-assisted pathogen is released intentionally or accidentally, it won't matter that 1,000 scientists have access to the model to design a cure.
It absolutely will matter. They'll solve it faster.
Think about the lag time, the time between designing a countermeasure, synthesizing it, clinically testing it, and distributing it globally would be measured in months or years. The damage would already be catastrophic. We must have institutional gating for generative biological models.
But that lag time you just mentioned, the months or years it takes to synthesize, test, and distribute, is exactly why the risk of a rogue actor creating an immediate pandemic is overstated. They face the exact same physical constraints. As we look at the reality of this science publication, we must ground ourselves in what actually happened.
Which is the creation of a generalized AI for biology.
Which is the creation of a historically significant, heavily vetted tool that opens a desperately needed pathway to personalized phage therapy. It took an incredibly simple, well-understood genome like Phi X-174, generated hundreds of thousands of sequences, and eventually yielded 16 functional viruses that solely target E. coli.
I know the stats.
The immediate, mathematically verifiable reality of antibiotic resistance far outweighs the theoretical, distant risks of bioterrorism, especially given the steep physical barriers to synthesizing viable pathogens today.
Look, I don't deny that the medical application here is profoundly important. I really don't. But we have to look past the specific E. coli experiment. Distributing a foundational model that natively understands the complex grammar of DNA fundamentally alters the landscape of global biosecurity forever.
You really think it's that absolute?
I do. As the underlying AI technology inevitably improves, and as the physical synthesis barriers lower with cheaper, decentralized lab equipment, Relying on the friction of physical bottlenecks is an unsustainable defense strategy. The safeguards can be stripped away, the models can be fine-tuned on dangerous data, and the capability to design living organisms has permanently left the controlled environment of institutional biology.
Well, it seems we are looking at the exact same data and drawing very, very different conclusions about where the locus of control should lie. But I think there is a profound point of convergence between our perspectives here.
Oh.
Yeah. We both unequivocally agree that this publication in Science marks a definitive paradigm shift in biology. Generative genomics has arrived. It is no longer a hypothetical concept. It is empirical reality.
Absolutely. I fully agree with that. The era of designing biology from scratch using artificial intelligence is here. And, well, we also agree that our current biogovernance models are entirely unprepared for this new reality. The gap between what AI can do in a lab today and what our institutions are prepared to monitor is vast, and it's growing exponentially.
Precisely. The tension between accelerating legitimate, potentially world-saving medical innovation and controlling the unprecedented biological risks of these models is only going to intensify as this technology evolves.
It really is.
It leaves us to ponder just how we navigate this new frontier. So we leave it to you to consider where that line should be drawn. When a machine finally learns the language of life well enough to write its own books, do we place it in the public square for all to read, or do we keep it securely locked in the library?
That is the million-dollar question.
Thank you for joining us.